All right, well, welcome everyone. So we're going to pick up here and begin discussing the activity and the events going on within our cardiac myocytes, right? So if you recall, the previous part we talked about our pacemaker potential, right? And essentially what that was doing is that was establishing and setting kind of the baseline heart rate, right? So each time we had a pacemaker potential, and again, remember those are occurring in those conductile cells in the SA node. So those those are the autorhythmic cells. What we'll do now is we'll essentially go beyond those events that we talked about in the previous part. So after the pacemaker potential initiates that signal and it sends that signal throughout the heart, what are then the cardiac muscle cells doing in response, right? So what are the cardiac myocytes doing? So we'll talk about the electrical activity there. We'll talk about the cardiac action potential, which will have some similarities to the neuronal action potential. And then we will talk a little bit about ECG. We may do ECG in a separate part on its own, right? So when talking about cardiac muscle cells, they have features that are very similar to the features that we see in other cells, right? So when we talk about other types of muscle cells, when we talk about neurons, right, there are different types of channels. Will involve sodium, will involve calcium, will involve potassium. And these will have a resting membrane potential, again, just like all of our other cells, right? So in this case, for our heart muscle, and again, this is the cardiac muscle, the cardiac myocytes, the resting membrane potential is going to be approximately -90, right? Recall, for our SA nodal cells, the resting potential was somewhere around -60, right? So slightly different from what we were talking about in our previous one. We're going to talk about what are called sodium channel, fast voltage gated sodium channels. We're going to talk about calcium channels. We'll discuss the role of potassium as well, right? So this is our cardiac muscle fiber or cardiac muscle cell, right? So remember, it has a resting potential. Again, in this case, it's going to be -90. Here's our sarcolemma. We have T-tubules. Remember, it's striated. We have all those kind of same basic features that we see in some other types of muscle tissue as well, right? So with skeletal muscle, right? So what we're going to do is we're going to kind of look through a series of events that's occurring in the cardiac muscle fiber, cardiac muscle cell. And some of these events will sound very familiar to you if you recall when we did our neuronal action potential discussion, right? Or whenever you've had that discussion in the past. So we'll talk about depolarization, we'll talk about repolarization. There'll be an extra phase that will add to the cardiac action potential, but many of them will at least sound somewhat familiar to you, or hopefully they should, right? So the first phase that we'll see is called the depolarizing or the depolarization phase, right? So this is occurring kind of while or right after we are at that resting membrane potential, right? So remember, resting membrane potential, we're at -90, okay, at -90. And then the cardiac muscle cells, right, they'll be stimulated from the activity coming from the SA nodal cells, right? So those SA nodal cells, again, they initiate the electrical impulse. It spreads throughout the heart, and then it ultimately stimulates our cardiac muscle fibers, right? It stimulates our cardiac muscle fibers. So in this case, we have voltage-gated sodium channels open, okay? Think about where sodium is highly concentrated, right? Sodium channels, or sodium itself is highly concentrated outside the cell. So whenever you open sodium channels, sodium is going to begin rushing in to the cell, right? So you have this influx of sodium rushing into the cell. And what that's doing is it's making the inside of the cell more positive. So again, remember that -90 is inside relative to the outside. So as sodium rushes in, it's going to increase the membrane potential from -90 up to around +30 or so, right? Depending on the text you look at, depending on the sources you're looking at, it may say +20, +30. We're going to go with +30, right? But just know that there may be some variation depending on the source that you are using, right? So that's this initial phase, and we'll look at a graph here in a moment. Depolarization, right? Sodium coming in, membrane potential becoming more positive, right? Once you get up to that +30 or so, you're going to begin to have the sodium channels close, so they're essentially becoming inactive, which is different than them closing, but we'll ignore that for now. And then you enter into a phase called the plateau phase, right? Now this is a phase that is unique to cardiac muscle tissue, right? It's unique to cardiac muscle tissue. Now, in the plateau phase is really preceded slightly by a brief repolarization. So you get slight repolarization. So in other words, a little bit of maybe potassium leaving the cell. And then you move into this plateau phase, right? We'll talk about the full repolarization phase next. But you move into this plateau phase, and plateau phase is characterized by the movement of two different ions, right? One is calcium and one is potassium, right? For this one, a little bit of potassium is leaving the cell and calcium is entering the cell, right? So potassium efflux, potassium leaving, and calcium influx, calcium coming in, right? So potassium going out, a little bit of calcium coming in, right? Potassium out, calcium in. What this does is it kind of causes the membrane potential, the voltage across the membrane, it causes it to remain relatively steady, right? So in other words, it kind of causes the membrane potential to become flat, right? Hence the name plateau, right? A plateau is this kind of flat region, right? So as the potassium is going out slightly, and then you have this increase in calcium permeability and calcium rushes in, you kind of get this balancing out, and you end up with this plateau phase. Now, this plateau phase, think about what this means for the heart's contraction, right? So when you think of depolarization, you can essentially think of depolarization contributing to the heart contracting, right? When you think of repolarization, and we'll talk about that in a moment, when you think of repolarization, you can essentially think of that as the heart beginning to relax, right? So this plateau phase is essentially kind of the phase in between, right? So the heart begins to contract, and before it relaxes in repolarization, it goes through this plateau phase, right? So think about what that contributes to, right? What does that mean for the heart's contraction, right? And we'll discuss a part of it here in just a moment, right? So again, potassium going out, calcium coming in. As calcium comes in, that's actually the stimulus for additional calcium release from the SR, from the sarcoplasmic reticulum. So remember, the sarcoplasmic reticulum in different types of muscle cells stores calcium, and so calcium influx is the stimulus for calcium release from the sarcoplasmic reticulum. That calcium release is then able, that calcium that is released is then able to go and bind onto troponin, and again, this is initiating our muscle contraction that we've discussed in previous lectures, right? So depolarization, plateau, and then we have our repolarization phase, right? So repolarization is now those voltage-gated calcium channels are closing. Calcium from the inside of the cell is being reabsorbed, kind of, and being pumped back into the sarcoplasmic reticulum. The potassium channels open. Potassium continues to leave. Potassium is highly concentrated inside the cell. So as the potassium goes out, it's taking with it its positive charge, and it's going to bring the membrane potential back down from that very positive value back down to our resting membrane potential of somewhere around -90 or so, right? Somewhere around -90, right? Now, and here, let me show you this graph. So this is the graph essentially showing the events that we just described, right? So again, depolarization, plateau, repolarization, right? Now, one important thing is the role of this plateau phase, right? The role of this plateau phase. And the plateau phase is essentially, it's going to contribute to a prolonged refractory period in the cardiac muscle cell, right? It's going to result in a prolonged refractory period in the cardiac muscle cell. What that means we'll discuss here in just a moment, but I want you to keep that in mind, that plateau phase, this middle phase right here. So again, potassium going out slightly, calcium entering, and no electrical change. So you can see how it's relatively flat, right? There may be a slight decline, but it's relatively flat. That plateau phase contributing to a prolonged refractory period. So keep that in mind, and we'll come back to that in just a moment, right? So when you're looking at these three phases, depolarization, plateau, repolarization, I want you to think of a few things. One, think of the ion that is involved, right? So what ion? Depolarization, it's sodium. Plateau, it's potassium and calcium. Repolarization, it's potassium. So that's one thing. Second thing is I want you to think of the direction that the ion is flowing with respect to the cell. So depolarization, we said it's sodium. Sodium is coming into the cell. So sodium influx. Plateau, potassium and calcium. Potassium coming in. Potassium leaving slightly. Calcium coming in. And then for repolarization, potassium, it's leaving. It's efflux, right? So that's the second thing. And then think about what's happening to the actual voltage of the membrane, right? So is it becoming more positive? Is it becoming more negative? For depolarization, it's becoming more positive. Plateau, think it's staying relatively steady, right? So no major change. And then repolarization, it's becoming more negative, right? So think about those two things: the ion, the direction the ion is moving, and the change in membrane potential for each of these, right? And you can see that up here in these three images, right? So what I mentioned just a moment ago is this idea of something called a refractory period, right? Now, a refractory period is essentially the time in which a cell cannot be stimulated again, right? So for something like a neuron, right, the refractory period is the time in which another action potential cannot be generated, right? For when you think of things like muscle, if you can have another action potential be stimulated, and that causes additional, contraction of the muscle cell, then that results in the cell essentially kind of combining and adding together multiple contractions into one larger contraction, right? And what it could potentially mean is that there's no relaxation in the cell. So the cell never relaxes, right? Now, for our skeletal muscle, our skeletal muscle can exhibit something called tetany, right? So this term up here, tetany. Tetany is a sustained contraction, right? Due to the short refractory period that you get in skeletal muscle. For our cardiac muscle, right? So for our myocardium, it cannot exhibit tetany, right? So in other words, this tetany, this sustained contraction, cardiac muscle cannot do that. Okay, it cannot undergo tetany. It doesn't exhibit tetany because it has this long refractory period, right? So in other words, after the heart muscle is contracted, or after it's stimulated to contract and it contracts, you can't add another stimulus to the heart that causes it to continue to remain contracted, all right? So what this is doing, again, it's making the sustained tetanic contraction impossible, right? Why is that important? Why is it important that the heart does not contract and then stay contracted, right? Why is it important that you can't combine or summate these electrical impulses onto the heart and cause it to remain contract? Well, if you think about the role of the heart and what it's doing, the heart is pumping blood. So in order to pump blood, the heart has to fill with blood. In order for the heart to fill with blood, it has to relax, right? It has to relax. That's the key. It has to relax. So how does the heart relax if it's in this sustained contraction? So in other words, it contracts and it just stays contracted. How does it relax? It doesn't, right? So to prevent that, we have this heart that has this prolonged refractory period. It's unable to undergo this tetany. So after it contracts, it then relaxes, it fills with blood, it contracts again, and it ejects that blood, right? So it's allowing for this filling, which then subsequently allows for this ejection of adequate and appropriate amounts of blood into the periphery, right? So the fact that the heart cannot exhibit tetany, that's a very good thing for our heart, right? That's a very good thing for our heart. Now, let me continue down here. So let me see, we were just on this slide. So this is a good example of what's happening in a graphic showing what's happening in skeletal muscle. Okay, what's happening in skeletal muscle. So notice here this image here on the left, right? So you get a single stimulus. Okay, so this blue line is showing the stimulus, the electrical impulse. So depolarization, repolarization, and so on, right? Notice here that the peak muscle contraction is occurring after this green region, right? So this green region is the refractory period. So in other words, during this green period, the neuron cannot send another impulse. Okay? It can't send another electrical impulse. However, the muscle is reaching its peak contraction after that, right? After that, there's no more refractory period, so it can get another stimulus, right? So you could stimulate the muscle again, and you can add onto it an additional contraction. So what this graph on the right is showing is that the tension, right? So this axis here is showing tension over time and the frequency of stimulation. You can add or summate these multiple contractions together because you get this, or you have the ability to have multiple impulses or multiple stimuli be summated together on the skeletal muscle, right? So in other words, big idea, big summary from this graphic here is that for skeletal muscle, you can keep a muscle sustained, you can keep the muscle contracted with minimal to no relaxation, right? This graphic here is showing the same type of thing, but it's showing it for cardiac muscle. And notice in this case, the green, this is the long refractory period, right? So again, here's the graphic we just showed, right, or that we talked about earlier. Here's depolarization, here's plateau, here's repolarization, right? Notice here that we're beginning to have muscle tension increase, and we reach peak muscle contraction during the refractory period, and we're almost completely relaxed when the refractory period ends, right? So what this is showing here on this right is that as the muscle is contracting and it's generating tension, right, you cannot add an additional stimulus, an additional impulse to the heart that results in a sustained contraction. So in other words, you get the relaxation. You get contraction, you get relaxation to a pretty good extent, and then you can have another stimulus. And then it can contract again and relax again, and this process can continue. But this is due to this really long refractory period, and this is due in part because of this plateau phase, right? It's due in part because of this long plateau phase, and then you get repolarization. Whereas with our our neuronal action potential, right, you have a very short refractory period, which allows multiple stimuli to be summated. You can add them together. All right. So we're going to stop here, and we'll pick up in our next part, and we're going to look a little bit at our EKG.